Hello, everyone, and welcome to today's webinar, a scalable miniature circular GenCircle dsDNA production system for gene and cell therapy. I am Cassie Saltman of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by GenScript. To learn more, visit genscript.com. This webinar is educational and thus offers free continuing education credits. Please click on the continuing education window at the bottom of your screen to obtain your credits after the webinar. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speaker, Dr. Fan Zhou, Senior Scientist at GenScript Novel Therapeutic Materials R&D Team. Dr. Zhou, you may now begin your presentation.
Hello, everyone. Thank you for joining us today on the webinar, a scalable miniature circular GenCircle dsDNA production system for gene and cell therapy. My name is Zhou. I'm a senior scientist of molecular biology at GenScript. Today, I'm going to spend about 30 minutes to overview the GenCircle dsDNA, including the following 5 aspects. The first is the background about Plasmid or circular dsDNA for GCT, and next is the introduction of GenCircle dsDNA development, and the next is the applications we can use the GenCircle to improve the performance or reduce the risk. And next is the manufacturing and QC specification, including 2 purities levels of the RUO grade services. And the 5th is how to design your constructs and customize your GenCircles. Plasmid has been commonly used as a vector system to provide genetic information in non-viral gene and cell therapy or DNA vaccine. Although much safer than viral approaches, plasmid vectors generally do encode antibiotic-resistant genes in backbone. These antibiotic-resistant gene markers, as well as the bacterial region, constitute a possible safety risk, and they are associated with structural plasmid instabilities and decrease gene delivery efficiency. These drawbacks have initiated the development of various mini-plasmid or circular dsDNA free of antibiotic-resistant markers. For example, the plasmid with a selection system based on the operator-repressed titration, or selection based on the RNA and RNA interactions, and the mini-intronic plasmid whose replication and selections elements are placed within an engineered intron in the eukaryotic expression system, and the famous mini-circle, devoid of any prokaryotic backbone, including the replication origin. So these novel classes of plasmid or circular dsDNA indicate a trend of plasmid development that is we want lower cell toxicity and lower immune response. So the backbone should have minimized size and free of CpG island. We want higher expression efficiency and longer duration, so we should eliminate the bacterial region to avoid transcription silencing. And it is also important to reduce biosafety risk caused by the use of antibiotics and its related antibiotic-resistant genes. And last but not least, the plasmid manufacturing should be easy to operate and easy to scale up and can meet the GMP quality requirements. Now I'd like to introduce a novel circular dsDNA system named GenCircle dsDNA and its production method. GenCircle dsDNA is a small circular DNA vector for gene expression or insertion. There is only 429 base pair, the size of vector backbone, contains mainly a replication origin. It is free of antibiotic-resistant gene or any other selection markers. There is no CpG island or bacterial region sequences in vector backbone. So it has many advantages over conventional plasmid. It can avoid regulatory concern for transgenetic drug resistance and has lower cytotoxicity and immunogenicity and have better stability and transfection efficiency. It also can increase knocking efficiency or viral titer or RNA and protein expression level. Now I will show you how GenCircle is produced. Firstly, we construct a specific parental plasmid. The plasmid contains selection marker and functional elements, as well as a pair of recombination sites. Next, host bacteria is transformed with the parental plasmid, and positive colonies are selected. Then we induce the expression of recombinase, and the self-recombination is occurred. The parental plasmid is separated into 1 molecular of GenCircle mini plasmid and 1 molecular of a circular selection marker with parental plasmid backbone. Then, because of the parental backbone circle is not able to replicate, it will be removed quickly during the bacterial growth. The GenCircle has a much higher copy number than parental plasmid, so GenCircle become dominant and soon replaced the parental plasmid completely due to the plasmid incompatibility. As a result, over 80% of the E. coli cells carry pure GenCircle and are without antibiotic resistance. It is verified by NGS analysis. Then single colonies are picked out for cell banking and plasmid fermentation. That is the basic mechanism of GenCircle production. GenCircle can be used in various applications in replace of conventional plasmids to eliminate the biosafety concern and improve the performance. It can be used in the cell therapy as the CRISPR knocking template or the transgene vector for the transposons, or it can be used in gene therapy as the material for virus packaging, or used as the non-viral gene of interest vectors, or as a material to protect the mRNA. It also can be used in DNA vaccine to express the antigen, and it can be used in the disease modeling and gene function study. Now let me show some details of case studies. The first one is the HDR knocking assay. We use the GenCircle as the template for HDR knocking. We compared the GenCircle with the traditional plasmid, for example, the PAC plasmid. It shows much higher knocking efficiency compared to the conventional plasmid. The knocking efficiency increased up to 30% compared to the conventional plasmid. And the other benefit is that no risk of transgenetic antibiotic resistance. And recently, we achieved a higher knocking efficiency by optimizing the transfection conditions. We observed as high as 57% of knocking efficiency using the GenCircle as a knocking template. And also, low cell toxicity was observed. And GenCircles can also be used in transposon systems. Now we show the piggyback transposon system using the GenCircle as the transgene vector. And we also compared with the traditional plasmid. It shows an increased efficiency up to 15% compared to the conventional plasmids. And in Sleeping Beauty transposon system, we also observed a high integration efficiency up to 45%. And GenCircle works well in both double plasmid system and the plasmid plus mRNA system. For in the application of virus packaging, the GenCircle can replace the conventional plasmid as the packaging vector or the transfer vector in lentivirus vector packaging and AAV packaging. We observed increased virus titer and no antibiotic resistance genes were detected in the virus particles. For the applications of in vivo or ex vivo gene expression, the GenCircle also showed a superior performance compared to the traditional plasmid. It shows an increased transcription level. And the transcription level increased fourfold compared to the conventional plasmid. This is a mouse assay that the mouse tail vein injection with naked DNA, and we monitor the RNA level after 9 hours after the injection. It shows fourfold higher than the traditional plasmid. The GenScript have the IP of GenCircle dsDNA as well as its related process. The complete manufacturing workflow include the gene synthesis, the parental plasmid cloning, and the GenCircle cell banking, and fermentation, and maxi prep and QC. In GenScript, we can provide GenCircle preparation services from gene synthesis to maxi prep within 2 to 8 weeks of turnaround time in total. And GenCircle systems show good compatibility with various lengths of inserts and complex sequences such as ITRs of AAV or long inverted repeats can be also produced stably. Besides sequence stability, GenCircle also shows superior homogeneity. Over 90% supercoiled monomer is detected in the final product. For the RU, the QC specifications are listed. For the RU grade GenCircle services, we provide 2 different purity levels for different assays and target cells. Then I will show you how to design your construct with GenCircles and customize your GenCircle product. As I mentioned, mentioned, GenCircle can be used in various applications such as gene expression, the HDR template, the transposons, and AAV or lentivirus transfer vector. So when you design your construct in gene expression, because the GenCircle, the backbone of GenCircle contains only replication origin or backbone. So you must include all the the functional elements for gene expression in your gene of interest. For example, the promoters, the ORF, the poly A, or terminator and the necessary UTR. For HDR knocking template, you should include the gene of insertion as well as the HA sequences in your inserts. For transposon applications, the ITRs of the transposon and the gene expression cassette for integration should be included. And next, for the AAV transfer vectors, the insert should include an ITR and the 4 cassette of transfer gene. And also for the lentivirus transfer vector construction, the LTR with functional elements and transfer gene should be included. Now we can provide the RUO GenCircle preparation services. As I just mentioned, the RUO grade of GenCircle services have there is 2 different purity level of industrial level and the preclinical level. The industrial level is recommended to be used for the viral vector packaging or the cell line test. And the preclinical level of the GenCircle has much lower toxicity. So it is recommended to be used in primary T cell or NK cell engineering or animal assays. And we also recommend to use the preclinical grade of GenCircle dsDNA in CRISPR HDR knocking assay. And we have observed a significant increase of knocking efficiency compared to the conventional plasmid. And we are also planning the GMP grade of GenCircles, and it is to be determined in the 4th quarter of this year for the IND applications and the early clinical trial. We provide from microgram to gram scale, and the optional delivery form such as the freeze-dried or the liquid, and the buffer is also optional. So that's all for my presentation, and thank you for your time with me today. Now, if you have any questions, I will be happy to answer them. Thank you.
Thank you, Dr. Zhou, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, how long does production take, and what is the expected turnaround time for an order?
The production takes from 2 weeks to 8 weeks in total. It depends on the length and the complex sequence in your GOI. Generally, the production of GenCircle takes only 3 additional 3 days of additional time for recombination and cell selection. Thank you.
Okay, next question. Does GenScript offer an animal-free production option for GenCircle?
Yes, we have animal-free production options as an add-on service. Thank you.
Great. Our next question. Can I perform the plasmid preparation myself?
Actually, because the GenCircle lacks the selection marker, so the E. coli cannot be transformed with GenCircle dsDNA, so it cannot be reproduced with only the GenCircle DNA. Thank you.
Okay, great. And it looks like we have time for one more question. What are the minimum and maximum insert lengths for GenCircle dsDNA?
GenCircle has a good compatibility with various lengths of inserts, but 1 kb to 8 kb is preferable. Thank you.
Okay, thank you, Dr. Zhou. Do you have any final comments for our audience?
Yes, I think if you are interested in GenCircle, you can feel free to contact me or my colleagues. We are very happy to share more details about it and help you with your essays. Thank you.
Thank you again, Dr. Zhou, for your time today and your important research. We would also like to thank LabRoots and our sponsor, GenScript, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.